Welcome back. Let's continue with our Week 6 lessons. In Unit 3, let's look at two very important methods. Let's first review the questions again. What features differentiate disease-causing missense SNVs from the neutral ones? How can we use these features to predict whether a missense SNV is disease-causing or not? Instead of working with nucleotide changes, almost all the prediction methods of missense variations work directly on the amino acid changes. Please take a moment to think, how would you predict the functional and phenotypic effects of an amino acid change? In 1999, Eric Lander's group made an early attempt at these questions in a part of their paper in Nature Genetics. They used the Blossom substitution matrix for the prediction, based on the assumption that if the substitution score between a variant residue and the wild-type residue is positive, then the variant is neutral. If the substitution score is negative, then the variant is deleterious. It was intuitive, but unfortunately didn't work very well because different proteins and different positions have very different tolerance for the same substitution. Since 2001, a number of more successful methods were developed. They were based either on sequence alone or sequence plus structure, and can be roughly divided into three categories: conservation-based, rule-based, and machine learning classifier-based. Let's look at them one by one. The best example of a conservation-based method is SIFT, for sort intolerant from tolerant substitutions, published in 2001 by Steve Hanikoff and colleagues. By the way, the Blossom matrices were also developed by Hanikoff's group. SIFT bets on evolution and aims to distinguish deleterious mutations from neutral ones. Its basic premises are that important positions such as active sites tend to be conserved across different species, so mutations at well-conserved positions tend to be deleterious. On the other hand, some positions tend to have a high degree of diversity across species. Mutations at these positions tend to be neutral. Thus, SIFT prediction is position-specific. So, how would you implement this nice intuitive idea computationally? SIFT takes a multi-step procedure. Given a protein sequence and an amino acid variation, in step 1, SIFT searches SwissProt database for similar protein sequences using 4 iterations of SciBlast with parameters E set to 0.0001 and H set to 0.002. SciBlast results that are over 90% identical in the regions aligned are grouped together, and a consensus sequence is made for each group. The motif method is used to find conserved regions and generate consensus sequences. Iteratively, more similar sequences are added until conservation in the conserved region decreases. Conservation at position C, RC, is defined using information theory as log 2 of 20 minus this sum over here. PCA is the frequency at which amino acid A appears in position C. The product of PCA and log PCA is summed over all 20 amino acids at position C. Step 2 extracts multiple alignment of the chosen sequences from SciBlast results. As you can see in this example alignment, some positions are highly conserved, whereas others are more variable. Step 3 converts the multiple sequence alignment into a position-specific scoring matrix and calculate normalized probability of seeing a particular amino acid in nature. For example, here we have the aligned amino acids at position C. Let's first define a few terms. NC is defined as the total number of sequences aligned at position C. GCA is the sequence-weighted frequency that amino acid A appears at position C. If there is a gap in the alignment, and if GC' is the frequency of gaps observed at position C, then for all 20 amino acids, GCA is increased by GC' divided over 20. Because the sequences included in the multiple sequence alignment is only a sample of all evolutionarily homologous sequences, we need to consider the possibility that some amino acids that exist in nature at position C in this protein, but they are not observed by us. So to deal with this, we introduce pseudocount FCA for unobserved amino acid A that might occur at position C using a 13-component Dirichlet mixture, which you can find more detail in the reference over here. Now, BC is defined to give the pseudocounts more weight relative to observed counts when the amino acids observed at a position C are more diverse. At an invariant position, BC is set to 0. At other positions, BC is defined as the exponential function of the sum over all 20 amino acids of the product of GCA with the rank RA of amino acid A among all amino acids in terms of BLOSSOM 62 similarity with the reference amino acid. So, the probability of seeing amino acid A at position C in nature is defined as the weighted average of the observed amino acid frequencies in this alignment and the estimated unobserved frequencies. This is then divided by the probability of the consensus amino acid at the position C to get the normalized probability of seeing an amino acid in nature at this position. If the normalized probability of an amino acid A is less than or equal to cutoff 0.05, it means that amino acid A is rarely seen at position C in nature. Thus, if we see that the wild-type amino acid is changed to amino acid variant A at this position in the protein, this variant A is considered damaging. Otherwise, it is considered tolerated. SIFT can be used online. You can input the sequence or genomic coordinate. SIFT will output a table like this, one for each variant. It will tell you whether it predicts the variant to be tolerated or damaging, and with what score. In their 2006 paper, Hanikoff and colleague reported that SIFT has a false negative rate of 31%, meaning that 31% of phenotype changing variations were wrongly predicted by SIFT to be tolerated. It also has a false positive rate of 20%, meaning that 20% of neutral variations were wrongly predicted by SIFT to be damaging. Because SIFT only works on positions that have at least 2 homologous sequences aligned, there are 40% of amino acid positions that SIFT cannot return a result. Thus, its coverage is 60%. Before we go on to the next method, let's stop briefly to look at the definitions of accuracy of a computational prediction method. The principles are the same for evaluating an experimental method. Assume that we want to know about certain truths about nature. For example, we want to know whether a variation may cause phenotypic change. Some variations indeed cause phenotype change, whereas other variations don't cause phenotype change. Suppose we have developed a new predictive method and we tested the method on variations that cause phenotype changes as well as variations that don't. The predictive method would predict some variations to cause phenotype change and predicts others to be not. We can then break them down into 4 different situations and fill in a 2x2 table. When we predict a variant to be phenotype changing while it really is so, we have a true positive case. When we predict a variant to be phenotype changing while it really is not, we have a false positive case. Similarly, when we predict a variant to be not phenotype changing while it really is not, we have a true negative case. When we predict a variant to be not phenotype changing while it really is, we have a false negative case. So how do we calculate the accuracy of our prediction method? The true positive rate, or sensitivity of our prediction method, is calculated as the number of true positive cases over the total number of positive cases. The true negative rate, or specificity of our prediction method, is calculated as the number of true negative cases over the total number of negative cases. The positive predictive value, or precision, is calculated as the number of true positive cases over the sum of the number of true positive cases and the number of false positive cases. Whereas the negative predictive value is calculated as the number of true negative cases over the sum of the number of true negative cases plus the number of false negative cases. And the accuracy is usually calculated as the sum of the number of true positive cases and the number of true negative cases divided by the total number of all cases. We also often speak of false positive rate, which is calculated as the number of false positive cases over the total number of negative cases. In statistics, it is also called a type 1 error, or alpha, and is equal to 1 minus specificity. Similarly, the false negative rate is calculated as the number of false negative cases over the total number of positive cases. In statistics, it is often called a type 2 error, or beta, and is equal to 1 minus sensitivity. Finally, the false discovery rate, FDR, is equal to the number of false positive cases over the total number of test positive cases, and is equal to 1 minus precision. If it is the first time you look at this table, you may find it very confusing. But really, after you studied it long enough, you'll find that the definitions are actually very logical. These calculations of accuracy can be applied to not only computational prediction methods, but also experimental tests. Now let's move on to briefly discuss a good example of a rule-based method named PolyPhen, or polymorphism phenotyping. It was first published in 2001 by Pierre Bourg, Shamil Saneev, and colleagues. PolyPhen predicts functional effects of amino acid variations based on both multi-sequence alignment and protein structure features. It is based on 3 presumptions. The first is the same as in SIFT, that amino acid variations at conserved positions are more likely to cause functional changes. The second presumption is that amino acid variations that affect the active sites, interaction sites, solubility, or stability of a protein are likely to affect protein structure. The third presumption is that changes in protein structure are likely to cause changes in protein function, which are likely to cause changes in phenotype. Here is a flowchart of PolyPhen. It takes as input a protein sequence and the position and amino acid identity of the variations. Then it calculates 3 types of features. It first calculates a set of sequence-based features of the substitution site. Then it calculates PSIC, which measures the likelihood of seeing the 2 amino acid variations. Finally, it calculates the structural features of the substitution site. Now, SIFT only calculates 1 feature, so it can simply apply a cutoff. But how can PolyPhen integrate the evidence in the 3 different types of features? Well, it uses empirical rules and predicts whether a variant is damaging or benign. More specifically, PolyPhen first generates multiple sequence alignment of homologous sequences and calculates sequence features and PSIC profile scores. Then it tries to get the 3-dimensional structure of the protein or use homology modeling to predict its structure. It then calculates the structural-based features of the substitution site, including whether the site overlaps with disulfide bonds, active sites, or important secondary structures, whether it is on the surface or inside of a protein, etc. Finally, it applies empirical rules that they derived from scrutinizing the sequence and structural features of variations with known damaging or benign effects. For example, if a variation happens at a functional or bond formation site, then it is predicted to be probably damaging. If a variation and the wild type have very small PSIC score difference, then it is predicted to be benign. The advantage of PolyPhen is that when protein structural information is available or can be built, it may achieve improved accuracy. However, when the structure is not available, it falls back to using multiple sequence alignment. More importantly, the rules it uses are empirical and rough. In 2010, Shamils and Eve and colleagues published a new version of PolyPhen that incorporated more predictive features and, more importantly, replaced the empirical rules with a machine learning classifier naive base. The classifier-based PolyPhen 2 showed improved performance over PolyPhen. Our group had also developed a classifier-based method named SEPRED. In the next unit, we will take a close look at SEPRED. See you then.